Good afternoon, Sir Kenneth and Ms. Ariska. My name is Virta Marcella from Biotechnology Cohort 25, and on today's video, I'm going to be presenting about my research study in regards to CRISPR-based gene editing as a possible strategy in treating Alzheimer's disease. Now, first and foremost, Alzheimer's disease is a neurodegenerative disorder, which is a chronic, irreversible condition in which there is a damage in the overall nerve system. And it's characterized by progressive loss of selectively vulnerable population of neurons. And this indicates that this loss of neurons will result in shrinkage of brain tissues and overall damaging the neural network. Alzheimer's Disease International predicted that AD or Alzheimer's disease would eventually grow into 78 million cases by 2030, and this indicated the overall Alzheimer's disease becoming a prominent case affecting elderlies, because studies shown that the overall growth of progressiveness of Alzheimer's is indicated, or the overall aggressiveness grows in accordance to someone's durations or the patient duration, indicating that aging has indeed becoming a high risk factor for AD prevalence. And Alzheimer's disease clinical characterization can be seen through accumulation of abnormal proteins. These abnormal proteins as including neuritic plaques and also neurofibrillary tangles. And this accumulation is resulted through the overall accumulation of beta amyloid peptides and abnormal phosphorylation of tau proteins. And this abnormal proteins accumulation further resulted in cellular vulnerability, and it making neural cells prone to distressful environments, and as a result, this exposure to the stressful environment causing damages to the overall neuron system, and the damages that occurs on neuron cells are irreversible because neuron cells unable to regenerate due to the cells being terminally differentiated in nature. But however, with the overall nature of Alzheimer's disease being a disease that heavily dependent on protein accumulation, it is somewhat possible to basically be coded, to be edited on its gene to prevent those protein accumulation. And here we welcome CRISPR-Cas9. CRISPR-Cas9 is a gene editing technology that originated from Leicester regularly interspaced palindromic repeats, that is CRISPR. And essentially with CRISPR, on its early discovery, it is once again a palindromic repeats that back then scientists don't really understand the overall functionality. But after several years on a different research done by different scientists or different people, it is shown that CRISPR can basically be found on two remote domains of life, and it is acting as a natural immune system for the organism. It is activated by sensing the presence of foreign DNA, and as a result, this activation of immune system allows scientists to basically locate the overall CRISPR sequences, and basically opens the possibility of targeting the desired gene sequences for further gene editing. And yes, CRISPR-Cas9 does open possibility for treating Alzheimer's with its overall technology in gene editing. However, large scales of clinical trial has not been implemented because due to the overall research having to face multiple hurdles, one of which is the hurdles of focusing on trying to basically target desired sequences, becoming the overall bottleneck of the research. And as such, currently the studies are trying to focus on targeting specific genes or molecules. And so as a result, for my research study, it is aimed to explore the potential of CRISPR-Cas9 application in specific gene targeting regarding Alzheimer's disease treatments. Now, for the overall methodology of this research studies, it utilized Boolean operator with the specific sequence of trying to filter out any literatures related to CRISPR-Cas9 technology implementation for targeting specific sequence on Alzheimer's disease as a treatment or medication or trial. Now, through the found literatures, I've also able to basically deduct the overall reasoning for Alzheimer's disease, one of which is the accumulation amyloid B peptide, as previously mentioned, which is derived from the amyloid beta precursor protein, APP, a transmembrane glycoprotein at neuronal synapses, regulating synaptic formation, repair, and also transport. And this APP protein undergoes two proteolytic pathways, that is the non-amylogenic pathway and also amylogenic pathway. And both pathways, differences lies on its overall end product, and also the enzyme that is responsible of the APP cleavage, that is with the non-amylogenic pathway utilize alpha secretase enzyme, while the amylogenic pathway utilize beta secretase enzyme. Now, through the non-amylogenic pathway, alpha secretase enzyme cleavage produce C80 fragments and also a N-terminal extracellular soluble APP domain, and with the C83 fragments is then being cleaved further by Y secretase enzyme and resulted in a AICD, or that is APP intracellular domain fragments, and also P3 peptide, and both end products are considered to be non-toxic, and as a result, non-amylogenic pathway are considered to be beneficial, as it supports the overall neurodegenerative modulation and neuronal health. Now, for amyloidogenic pathway, it actually utilizes beta secretase enzyme, which the cleavage is done on the end side of the alpha beta domain, or the amyloid beta peptide domain. As you can see on the non-amylogenic pathway, the cleavage was actually done inside the domain. However, for the amylogenic pathway, it was done on the end side. As a result, it produced an SAAP beta fragment and also C99 fragments. And as a result, further cleavage by C secretase enzyme resulting in, once again, yes, it does produce AICD, the amyloid, the APP intracellular domain, but also leaving out, or basically creating the formation of amyloid beta peptide. And this formation of amyloid beta peptide will be accumulated or forming into oligomers and resulted in overall a larger protein plaque, that is the neurotoxic plaque formation, and which inhibits the overall transportation of neuronal signals to the brain. Other causes for Alzheimer resulted from the abnormal phosphorylation of tau protein, and tau protein is a microtubule associated protein known as a component of the neuron's skeletal structure, primarily localized at axons, and its regulation is done through phosphorylation, regulated by two types of enzyme, that is kinase, becoming the on switch for tau protein by allowing the transfer of phosphate group, and phosphatase, which actually removes the phosphatase group, becoming the off switch. as you can see through the figure, the healthy neuron have a somewhat balanced concentration of both kinase and phosphatase enzyme. However, for the Alzheimer's disease, when the neuron cell is having Alzheimer's disease, actually the tau protein have an imbalance in chemical concentration, with kinase favors heavily more on the overall system, and as a result, the tau protein are being hyperpolarized, or not hyperpolarized, but hyperphosphorylated, and as a result, it causes the tau protein to lose its overall affinity and lose its binding, and causing a disarray in the microtubule structure. Now, with both reasonings of Alzheimer's disease, we go back to CRISPR-Cas9, which is a revolutionary gene editing technology updated from the immune system in bacteria. And with this specific CRISPR-Cas9, there is a possibility to basically re-engineer the overall reasoning of the protein accumulation in hopes to basically prevent Alzheimer's disease. And for CRISPR-Cas9, it requires two types of key molecules, that is the guide RNA, gRNA, and also Cas9 enzyme. Now, the overall method for implementing CRISPR-Cas9 started with the injection of gRNA to match with the target DNA. And after injecting and also introducing the gRNA to the targeted DNA sequence, it will then become a somewhat locator or for Cas9 enzyme to actually goes, allowing the Cas9 to be led by the gRNA to the specific targeted DNA sequence. And after leading the Cas9 enzyme will basically scan for PAM, or protospacer adjacent motif, and by scanning this specific PAM system, it enables to basically apply its function to cause conformational changes on the DNA sequence. That is, the Cas9 will basically create double-strand break on the DNA sequences. Now, after creating the double-stranded break, the cells would naturally do DNA repairing system. That is the non-homologous end-joining and also homology-directed repair. With the non-homologous end-joining, are much more prone for causing indels or insertion or deletion to disrupt and enables to disrupt or disable the target gene, as this specific repairing system just basically connect two broken strands of DNA. However, if we actually provide a template for its repairing, we can do homology-directed repair, as the additional provided added template able to allow the DNA to basically fix itself by seemingly insert precise edit or gene correction on the broken strands. Now, the overall technology of CRISPR-Cas9 are basically being targeted on several enzymes and also several materials. For example, one of them is apoprotein E4 or ApoE4. ApoE4 is a enzyme that circulates in the bloodstreams of brain and sources by astrocytes, and this enzyme mediates the co-deposit process of amyloid beta peptides, and co-deposit refers to the removal of plaque formation, which lowers the risk of AD. So hypothetically, by targeting this specific gene editing on ApoE4 in hopes that it would basically lowers the overall alpha beta plaque formation. However, it actually resulted in several side, side effects that is show that the overall that the overall editing of it causes a lower affinity with its binding to alpha beta peptide and actually promotes the overall the amyloid beta formation or amyloid beta peptide formation, plaque formations. And other than ApoE4, we also have base 1, which is beta site amyloid precursor protein cleaving enzyme 1. This specific enzyme actually initiates the protein plaques formation. So, hypothetically, by targeting this specific enzyme, we could basically engineering it or compute it to lower down its overall concentration to not promote the amyloid beta peptide formation. However, targeting this specific base 1 enzyme, it would actually cause several side effects with the overall elimination of amyloid beta peptide would actually become somewhat toxic or become somewhat harmful for the overall neuron system. And as such, base 1 is discontinued due to ethical reasons. That is the overall side effects that is produced from base 1 targeting. And finally, we also have APP or amyloid beta precursor protein, which is the main derivative of amyloid beta peptide formation. That is the specific protein that is being cleaved by several enzymes, and with the hopes of targeting this APP protein to prevent the amyloidogenic pathway or the pathogenic pathway. And however, by resulting APP, it actually adverts cognitive side effects. One of them is the inhibition of the iron regulatory protein 1, which is an enzyme that is functioning to decrease neurotoxic protein and improve the overall axon integral structure. And also, once again, having the same problem with base 1, fully diminishing or fully eliminating any amyloid beta peptide production would actually be somewhat fatal for the neural system, as amyloid beta plaques actually helps in improving neuronal survival. So once again, APP is currently still in undergoing multiple research to see its opportunity to basically able to be used for further treatments. And as a result, in conclusion, despite being in early stages of Alzheimer and its application on CRISPR-Cas9, it opens up a promising possibilities for treatments and for advanced specific gene targeting technology. Because yes, despite all of the limitation that we face through different gene targeting, there is a possibility for it to actually be a cure or treatment for Alzheimer's disease. However, several limitations can be found, such as the delivery bottleneck. That is, the transportation pathways are the biggest hurdles. The reason that there is this specific molecule are being targeted is due to the overall duration of research and experimentation and trials that is needed to basically found any types of enzyme or molecules to be targeted requires time once again, and becoming the biggest hurdle. And there's also risk of cognitive decline with targeting specific molecules, specific enzymes. Obviously, will have different sides of effects, and it would potentially able to decline in overall the neuron functionality. And as such, more trials is required for this overall research. And as such, future research directions suggest that we should shift focus of targeting the specific gene sequences on neighboring molecules. So rather than focusing on molecules that are actually related fully on the Alzheimer's disease, maybe there will be some enzyme or other cells that are not related to the Alzheimer's disease but have the potential to actually decrease the overall protein protein accumulation. And that's going to be the end of my presentation regarding the overall regarding the overall exploration of CRISPR-Cas9 as a possibilities of targeting gene sequences as a cure for Alzheimer's disease. And once again, thank you for listening to my presentation. I am deeply sorry. I also want to deeply apologize for any mispronunciation or any words that happens to be somewhat misleading and also incorrect throughout my presentation. And finally, for AI usage declaration for this specific presentation is that AI were indeed used, but only on fixing grammars and also any typos that are found in the presentation text. And once again, thank you for listening to my presentation.